Hello everyone, welcome back to Biotech Hub. We are continuing with our bioinformatics series, and in this series we are going to talk about bioinformatics with context with various biological systems. So basically we are going to understand how bioinformatics is contributing towards different biological systems. Let's start with our very first system, that is nucleotide sequence. So basically we are understanding how bioinformatics is acting or is contributing towards understanding this nucleotide sequence. First, the nucleotide sequence storage. When we are talking about storage, we are basically talking about all the sequence storage that is occurring, that is present throughout. So we have all the things stored in a single format, in a single database. Second comes the organized form. Organizing, basically we are organizing the nucleotide sequence in a very specific way, so that whenever we want to get any nucleotide sequence, we can put in the command. We can put in the specifications, that these are our specifications, and we get that nucleotide sequence. So we can organize the nucleotide sequence in a very specific way, whatever specifications we want to give. Third comes the very important aspect, that is analysis. So when we are talking about analysis of the sequence, we are talking about a number of factors. Like we want to study a interaction between a nucleotide and a nucleotide. We can easily study that with the help of nucleotide sequence. If we know the sequence, we can understand, or we can analyze that what will be the interaction between a nucleotide and another nucleotide. Or we can also study the interaction with other components that are present. Like say suppose we want to understand the interaction of nucleotide, a very particular nucleotide sequence, with a particular protein, so we can understand that interaction as well. Moving forward, with this analysis only, we can study the content of that nucleotide sequence. Say suppose we have a sequence A T G C T C A. So in this sequence now, it's a very short sequence. So in this sequence we want to calculate what is the AT content, or we want to know what is the GC content of that particular nucleotide. So we can easily analyze. Now this is a very small sequence. When we are not talking about nucleotide sequence, we are talking about maybe 10 raised to the power 5 base pairs, to many more, maybe to the 10 raised to the power 9 base pairs we are talking about. So for that much amount of base pairs, we cannot manually calculate that how much amount of AT or how much amount of GC is present. So for that we are requiring bioinformatics. There come the properties of that nucleotide sequence. Say suppose I want to get the melting temperature. So I can understand the melting temperature very easily with the help of the sequence. If I have the sequence, I can calculate the amount, the number of nucleotides present. We can calculate how much energy will be required to break one of the nucleotide to base. Basically we can calculate the melting temperature for two nucleotides and for the entire sequence as well. So we can get to know the properties such as melting temperature, or maybe denaturation property, and many more properties. Next, moving forward, we have nucleotide structure. So how bioinformatics is contributing to nucleotide structures? uh, Knowing the base pair energy. So we want to calculate the base pair energy. We can easily calculate with the help of bioinformatics. See, if we want to calculate the base pair energy for a very short sequence like ATG, this is our sequence, we can easily calculate it. But if the sequence is of 10 raised to power 3 base pairs, we cannot calculate the base pair energy quite easily. So for such as for big data, we are using bioinformatics, or in general for the data we are using bioinformatics. can know the interactions we have already talked about, so we can understand interactions. We can also make database for nucleotide structures. For every biological system we can make a database. So making a database is also a kind of contribution that bioinformatics is doing towards biological systems. We can understand hydrogen bonding, how A is hydrogen bonded with T. There are two hydrogen bonds present. How is G3 hydrogen bonded with C? So these all interactions, hydrogen bonding, base pair energies, we can easily understand with the help of bioinformatics. Next come RNA or express gene. So when RNA and express gene, how bioinformatics is contributing, we can translate that sequence. When we know that the mRNA is translated to form proteins. So with the RNA sequence, we can easily analyze, or we can easily get to know the translated sequence, that what all sequence is getting translated and what are the transcribed amount in that particular RNA. Because we know that not the entire RNA is getting translated, only the coding region or the ORF region is getting translated from start to stop codon. So we can also predict the transcribed amount, how much amount is going to be we can also predict the transcribed amount. We can get to know about the introns and exons. We know that not the entire RNA is going to translate it into protein. We can understand somewhat, we can correlate with this intron and exon sequences. Moving forward, let's talk about protein sequence, how bioinformatics is contributing with understanding of protein sequences. So for protein sequence, we can get to know about the post-translational modifications. We all know that once a protein is transcribed from a RNA, we had a RNA sequence, we get a protein. So this protein undergoes few modifications. So we can get to know about these modifications by knowing the protein sequence. Next, we can do structural prediction. Once we know the sequence of the protein, we can easily predict what structure is going to form. Like whether it's going to be an alpha helix form, or whether it's going to be a beta sheet form, whether there will be a hinge region present between or not. All these things can be predicted just by knowing the protein structure. We can do some functional hypothesis. By knowing the structure, we can easily predict what kind of function or what type of function that protein is going to perform. So just by knowing the sequence, we can have a functional hypothesis about that particular protein. We can calculate the composition also. When I'm talking about calculating the composition, we are basically talking about calculating composition of a particular amino acid. Like if we want to calculate a particular amino acids concentration, say suppose I want to know how much glycine is present in that particular protein sequence. We are given a very long sequence of AGVL, G, M, D. So this kind of sequence we are given, and we want to calculate the G, that is amount of glycine present in that particular sequence. So what we can do, we can calculate that composition just by putting a very easy formula, that is occurrence of that residue that we want to calculate the composition for upon the total number. So this particular formula can give us the composition of that particular residue. Moving forward, we'll discuss this composition formula in depth. Moving forward, how bioinformatics is contributing with protein structure. So for protein structure, we can get to know a couple of things, like we can do some functional prediction. If we have the given structure in the alpha form, we can easily predict what kind of function is this going to perform in that particular region. So we can have some functional prediction based on the type of structure that we are given. We can do some study regarding the stability of that particular protein, that how much stability that particular protein holds. Say suppose I have a alpha helix structure, and I want to calculate that how much temperature this alpha helix can hold. So for that kind of stability study, protein structure is quite useful in bioinformatics. Next, we have residue location. So if we want to locate the residue location, say suppose this is my protein structure that is formed. So and this was the original sequence. So there was a glycine at number 10, and there was a valine at number, suppose, 100. So now when the structure is given, we can see where that particular residue is being located. Suppose this 10 number glycine is interacting with valine. So we can get to know about the locations of particular amino acids in that structure. Moving forward, if we are dealing with bioinformatics and protein interactions, so protein interaction can give us a couple of information, like how residues are interacting with each other. Just similar to this residue location itself, how these residues are interacting with each other, on what context, like, or even if we are talking about protein interaction, we are not just talking about a single protein, we are talking about a protein protein interaction, we are talking about a protein DNA interaction, and many more kind of protein interactions we are talking about. Interaction of protein with all the other components. So how these residues, which particular amino acid is interacting with that particular compound, we can get to know about that through this protein interaction study. We can get to know where the binding sites are present, or what particular amino acid pockets form that binding site. We can get to know the pattern of interactions, like what is the binding affinity, how it is binding, and all. This is a much depth of topic which we can discuss in our further lectures. Thank you all for your patient ears. Please like, share, and subscribe, and stay connected with Biotech Hub. See you all for the next one.